Hi, my name is Jelena Skorik, and topic of my presentation is an overview of the glycolysis and glycogenesis pathways. Within this presentation, I will define what glycolysis is. I'll talk about all the steps. I will also define what glycogenesis is and also talk about those steps. So what is glycolysis? Glycolysis is the breakdown of glucose into pyruvate molecules, ATP molecules, and NADH, and all this takes place in the cytoplasm of the cell. As you can see here, there's a lot of steps. Some of them are not even listed here, so it looks like a very complicated process, and that's why glycolysis is broken down. All the steps are broken down into 3 stages. So let's look into those 3 stages. So we have stage 1, that's called investment stage, and we'll see later why. It consists of 3 steps. Stage 2, cleavage stage consists of 2 steps, and third stage, energy generation stage or payoff stage, that consists of 5 steps, and that's the most complex stage. The reason we break down glycolysis into these 3 stages is because each one of these stages actually carries out its own specific purpose. So let's look into first stage, that's called investment stage. And the point of this stage is to take that glucose molecule and trap it inside the cell so that it cannot leave, and prepare it for cleavage, which takes place in the stage 2. So in the step 1 of this first stage, glucose makes its way into the cytoplasm within the cell, and the enzyme hexokinase takes phosphoryl group from the ATP molecule here and adds onto the carbon number 6. So we form glucose 6-phosphate. We break down ATP molecule into ADP molecule, and also the hydrogen ion is released as well. Now the point of this reaction is to first destabilize the glucose to make it more reactive and begin preparing it for stage 2. And second point is by adding this polar component here, we trapped glucose in the cell, so it will not be able to exit the cell because it cannot pass the membrane, and it cannot use any of those transport membrane proteins because its structure is different. In step 2, the goal is to take that glucose 6-phosphate molecule and transform it into an isomer into fructose 6-phosphate, because in stage 2, we basically want to produce 2 identical 3-carbon molecules. And so to produce those 2 identical 3-carbon molecules, we have to have symmetry in our molecule. So this is not symmetric, but this is symmetric, and so the glucose 6-phosphate is transformed into glucose 6-phosphate to make sure we get those 2 identical 3-carbon molecules in stage 2. This is an isomerization reaction because we transform one isomer into another, and the enzyme that catalyzes this reaction is called phosphoglucose isomerase. This is an exergonic reaction. It takes place spontaneously under physiological conditions. Let's look now at the step 3. The point of step 3 is to continue destabilizing this molecule. So in step 1, we destabilized it and increased its energy and made it more reactive because we made it more polar by adding a charge. And here we add a second charge and that makes it even more reactive and more likely to actually undergo cleavage in stage 2. So we take fructose 6-phosphate, and again, because we want to add the phosphoryl group, we are going to have phosphoryl fructose kinase. The process adds phosphoryl group onto this oxygen here. And now we have a symmetrical molecule. And once the cleavage takes place in stage 2, that will ultimately allow us to produce 2 3-carbon molecules. Because we are essentially investing to prepare that molecule for cleavage, we actually use in these steps of the stage 1, we use ATP, 2 ATP molecule. This stage is called investment stage. So now we are into stage 2, which is called cleavage stage, because this is where we break down this molecule here that we formed in stage 1. So we take the fructose 1,6-biphosphate and under the guidance of an enzyme called aldolase, we form these 2 3-carbon molecules. One of them is DHAP, which stands for dihydroxyacetone phosphate, and the other one is GAP, which stands for glyceraldehyde 3-phosphate. Once we form this GAP molecule, it doesn't do anything else, but DHP doesn't lie directly on the path of glycolysis, and so we have to take this molecule and transform into this one, which is its isomer. The enzyme that catalyzes this reaction is triose phosphate isomerase. So the net result of stage 2, we took fructose 1,6-biphosphate and we cleaved it into 2 identical 3-carbon molecules, these GAP molecules. So now we have stage 3. So essentially there are 3 parallel stage 3 reactions, because at the end of stage 2 we created 2 GAP molecules. In stage 3 we're going to produce those ATP molecules and pyruvate molecules. So the entire goal here is to destabilize the molecule and eventually create a molecule that contains a high potential to transfer phosphoryl groups, and we'll see why that's important in just a moment. So let's take a look at step 6. So we want to transform this GAP molecule into 1,3-biphosphoglycerate. Why do we want to do that? Because we want to transform a molecule that has a relatively low potential, which is GAP, to transfer phosphoryl group into a molecule that has relatively high potential to transfer that phosphoryl group. And so we basically take the GAP and mix with NAD+, and we also use an orthophosphate, and in the presence of GAP hydrogenase So the hydrogenase basically means we're going to have a reaction which there will be a transfer of a hydride group. And so this will reduce into NADH. We're going to release this hydrogen ion, and that orthophosphate from here will basically attack this molecule here and bind onto this carbon right here. And so now we have these two phosphate groups, this one here and this one right here, on this molecule. So one on the first position and one on the third position. And that is why this molecule is essentially a molecule that has a higher potential to actually transfer these phosphoryl groups. So now in the next step, we can use 1,3-BPG molecule to basically transfer that phosphoryl group onto an ADP molecule and produce an ATP molecule. And the molecule that catalyzes this will be phosphoglycerate kinase. So we mix 1,3-BPG molecule with ADP molecule. This will accept, which will accept this phosphoryl group from here. And so once the process takes place, we essentially form an ATP molecule and we form 3-phosphoglycerate. Now what happens with this 3-phosphoglycerate molecule? We'll see in the next step. In step 8, we basically want to transform the 3-phosphoglycerate into less stable molecule, 2-phosphoglycerate. So we want to take this one and make it more reactive so that in step 9 it can actually react. So in order to destabilize this molecule here, we want to take this phosphate and bring it closer to this negative charge. So we have negative charge, charge -1, and this charge -2. And if we basically decrease the distance between the negative charges, that will destabilize this molecule, and so we have an enzyme known as phosphoglycerate mutase, which will take this group here and move it from carbon number 3 to carbon number 2. And so now we have 2-phosphoglycerate, which is a lot more reactive than 3-phosphoglycerate. Now that we have this less stable molecule, they can react in step 9 and it can be transformed into a molecule that prepares it for the pyruvate. So we take 2-phosphoglycerate and we use enolase to transform it into phosphorenolpyruvate or PEP. We essentially form double bond between these, and H and OH combine into water molecule that's being kicked off. And so this is dehydration reaction. Once this molecule is not very stable and it has a very high phosphoryl transfer potential because this is essentially trapped into enol state, and it's trapped because oxygen, this oxygen here doesn't have an H, it has this phosphoryl group, And so what must happen in the final stages, phosphoryl group must be donated to ADP molecule and replaced with hydrogen ion. And once it's replaced with hydrogen, it can transform into more stable ketone state, the pyruvate molecule, and that's exactly what happens in the next and final stage. So in the final stage, we take this molecule here that is high in energy, so it contains very active bonds, and that makes it very good molecule that actually transfers that phosphoryl group onto ADP molecule in the presence of pyruvate kinase and forms pyruvate molecule and ATP molecule. And because this process takes place twice, we form two ATP molecules here, and we already formed two before, so a total of four ATP molecules. We use two ATP molecules in stage one, and the net result is the two ATP molecules in glycolysis per glucose that we actually use up. So if we sum all these individual reactions, keeping in mind that all these take place twice, we get the following net results. So we have a glucose molecule, we have two ADP molecules, we have two NAD molecules, plus two phosphoryl groups, and during the process of glycolysis, we create two pyruvate molecules, two NADH molecules, two ATP molecules, and we also have as a product two molecules of water and two hydrogen ions. Now let's take a look at glycogenesis. What is glycogenesis? Glycogenesis is the synthesis of glycogen molecules from glucose. So let's suppose we just ingested a meal that is rich in sugar molecules, and so what that means is inside our blood plasma there will be an increase in the glucose levels. Now our liver is responsible for maintaining the proper glucose levels of our blood. So what the liver cells do is they begin to uptake some of that glucose into the cytoplasm of the cell. Once glucose is in the cell, the liver cells will want to trap that glucose inside the cell and prevent it from actually escaping back into the blood plasma. So the liver cells phosphorylate the glucose molecule on the 6th position. This is glucose molecule. So it phosphorylates on the position 6. And they use an enzyme hexokinase. They take phosphoryl from ATP molecule and places onto the carbon 6 of glucose, and that creates glucose 6-phosphate. Now, once glucose 6-phosphate is created, it undergoes a reaction in which there is a transfer of phosphoryl group from the 6th position, so from this position, to the 1st position of that glucose, and we form glucose 1-phosphate. The enzyme in this reaction is phosphoglucomutase. In the next step, glucose 1-phosphate needs to be activated because by itself glucose 1-phosphate cannot simply be added onto the growing glycogen chain because it's not active enough. So we have to make it much more reactive. So we attach another phosphoryl group as well as uridine molecule or UDP to create UDP glucose. That takes place in this step 3, so we take the glucose 1-phosphate, shown here, and uridine 3-phosphate, or UTP, in the presence of enzyme known as UDP glucose phosphorylate, and create UDP glucose and the phosphate right here. The purpose of this was to create this high energy bond that is very active, because as we'll see in just a moment, this is able to actually undergo the reaction in which we'll attach it onto the growing glycogen chain. So once we have a bunch of these UDP glucose molecules, the enzyme glycogenin uses the UDP glucose molecules to create a primer, which is a short sequence of glucose molecules that are linked by 1,4 glycosidic bonds, because glycogen synthase need the primer to begin the process of extending that glycogen. So now we take UDP glucose in the presence of the primer. The enzyme glycogen synthase basically attaches this glucose onto the primer, and in the process we form also UDP molecule. Now once we continually add these glucose molecules, we want to start branching them, because that increases solubility of glycogen. And so that's where the enzyme glycogen branching is responsible for creating those alpha-1,6 linkages, which lead to the branching points. For this reaction to actually continue taking place, we need to take this UDP that we created in previous reaction and generate back the UTP molecule. That's where this step takes place. The enzyme nucleoside dephosphokinase uses ATP molecule to attach it onto ADP molecule and create UTP molecule and ADP molecule. Once we generate the supply of UTP, this process continues and we can continually build that glycogen polymer. And here, this is like a little picture where we see branched glycogen molecule. This would be it for this presentation. Thank you for your attention.